Introduction to microbiology. The first simple forms of life appeared on Earth more than 3 billion years ago. Their descendants have changed and developed into the several millions types of animals, plants, and microorganisms recognized today. Of course, thousands more remain to be discovered and officially described. Microscopic forms of life are present in vast number in nearly every environment known. That is soil, water, food, air, etc. Since the conditions that favor the survival and the growth of many are the same as those under which people normally live. Many microscopic microorganisms or microbes occur as single cell. Others are multicellular, and still others, such as viruses, don't have a true cellular appearance. Some organisms, called anaerobes, are capable of carrying out their vital functions in the absence of free oxygen, whereas other organisms can manufacture the essential compounds for their physiological needs from atmospheric sources of nitrogen and carbon dioxide. Other microorganisms, such as viruses and certain bacteria, are totally dependent for their existence of the cells of higher forms of life. The branch of science known as microbiology embraces all of these properties of microorganisms and many more. One of the attractive features of microbiology is a number of the investigation and work remain to be done. Many decisions affecting the future of the world may depend upon the involved activities of microorganisms in areas like food production, pollution control, energy production, and the control and the treatment of diseases. In short, microbiology has assumed a position of great importance in modern society. Medical microbiology, from Greek words meaning micros, small, bios, life, logos, science, is the study of causative agents of infectious disease of human beings and their reactions to such infections. In other words, it deals with the etiology, pathogenesis, laboratory diagnosis, treatment, epidemiology, and control of infection. Obviously, medical microbiology has a close link with the other disciplines, that is, pathology, clinical medicine, surgery, pharmacology, therapeutics, and preventive medicine. Microbiologists are enthusiastic to confirm the diagnosis and cause of macroscopic changes by taking smear and preparing culture from the lesions to demonstrate microorganism. Example, circumscribed boil of staphylococcus, the spreading cellulitis of streptococcus, the red liver, like appearance of lung in pneumococcal pneumonia, the tubercles followed by syphilis and intestinal ulceration of salmonella organism, etc. The pathology of infection is quite fascinating as it includes affinity of pathogens for certain tissue and initiation of infection and characteristic tissue reaction. Each investigation is regarded as a research project in many nature. Historical events. Long before the discovery of microorganisms, certain processes caused by their life activities, such as fermentation of wine, juice, milk, yeast, etc., were known to mankind. In ancient times, at the beginning of civilization, man used these processes there to prepare commerce, source milk, and other products. Antonio van Leeuwenhoek, 1683. He was the first scientist who observed bacteria and other microorganisms using a single lens microscope constructed by him, and he named the small organism as little animalcules. Robert Hooke, contemporary of Leeuwenhoek, developed compound microscope in 1678 and confirmed Leeuwenhoek's observation. Edward Jenner, 1796, developed the first vaccine of the world, the smallpox vaccine. He used the cowpox virus variola vaccine to immunize children against smallpox, from which the term vaccine has been derived. The same principles are still used today for developing vaccines. Louis Pasteur, microbiology developed as a scientific discipline from the area of Louis Pasteur, 1822-1895. He is also known as father of microbiology. He was a professor of chemistry in France. His study on fermentation led him to take interest to work in microbiology. His contributions to microbiology are as follows. Number one, he had proposed the principles of fermentation for preservation of food. Two, he introduced sterilization techniques and developed steam sterilizer, hot air oven, and autoclave. Three, he described the method of pasteurization of milk. He had also contributed for the vaccine development against several diseases such as anthrax, fowl cholera, and rabies. 5. He disproved the theory of spontaneous generation of disease and postulated the germ theory of disease. He stated that diseases cannot be caused by bad air or vapor, but it is produced by microorganisms present in air. 6. Liquid media concept. He used nutrient broth to grow microorganisms. 7. He was the founder of the Pasteur Institute, Paris. Joseph Lister, 1867, is considered to be the father of antiseptic surgery. He had observed that post-operative infection were greatly reduced by using disinfectants such as diluted carbolic acid during surgery to sterilize the instruments and to clean the wounds. Robert Koch, father of bacteriology. Robert Koch provided remarkable contributions to the field of microbiology. He was German general practitioner, 1843-1910. His contributions are as follows. Number one, he introduced salt media for culture of bacteria. Elishmans, he is the wife of one of Koch's assistants, had suggested the use of agar as solidifying agents. 2. He also introduced methods for isolation of bacteria in pure culture. 3. He described hanging drop method for testing motility. 4. He discovered bacteria such as anthrax bacilli, tubercle bacilli, and cholera bacilli. 5. He introduced staining techniques by using aniline dye. 6. Koch phenomenon. Robert Koch observed that guinea pigs already infected with the tubercle bacilli develop a hypersensitivity reaction when injected with tubercle bacilli or it is protein. Since then, this observation was called as Koch phenomenon. 7. He also suggests criteria before plumbing the organism responsible for disease. It goes by the name of Koch's postulate, according to which: number one, the specific microorganism should be isolated from all cases of specific disease and should not be found in healthy individuals. Two, the specific microorganism should be isolated from the diseases individual and grow in pure culture on artificial medium. Three, the isolated microorganism should reproduce the specific diseases when inoculated into healthy individual. Four, the specific microorganism should be re-isolated in a pure culture from the experimental infection. In addition, fifth criterion was introduced subsequently with the status that antibody to the causative organism should be demonstrable in the patient's serum. Exceptions to Koch's postulates. It is observed that it is not always possible to apply these postulates to study all the human diseases. There are some bacteria that don't satisfy all the four criteria of Koch's postulates. Those organisms are: A. Microbacterium, Libra, and Tierbonemia balanidium. They cannot be grown in vitro. However, they can be maintained in experimental animals. Neisseria gonorrhoeae. There is no animal model. However, it can be grown in vitro. Koch postulates. Lord Lister, 1854, used carbolic acid spray on wound during operation. He is also called father of antiseptic surgery. Neisser, 1879, described gonococcus. Augustine, 1881, discovered staphylococcus. Loeffler, 1884, isolation diphtheria bacillus. Loeffler and Froch, 1898, observed that foot and mouth disease of cattle was caused by microbe, that is filter passing virus. Walter Reid, 1819, observed that yellow fever was caused by filterable virus and that it was transmitted through the bite of mosquitoes. Landsteiner and Popper, 1909, show poliomyelitis was caused by filterable virus. Tort, 1951, and Herald, 1970, discovered lytic phenomenon in bacterial culture. The agent responsible was termed as bacteriophage viruses, group of viruses that attack bacteria. Alexander Fleming discovered penicillin in 1928. He made the accidental discovery that fungus Penicillium produce a substance which destroys staphylococci. Rosca, 1934, introduced electron microscope and hence detailed study of microbiology, morphology of virus was possible. New agents of infectious disease continue to emerge. Example, HIV, identified in 1980. The outbreaks of plague in 1994. Cholera, 1995. And dengue hemorrhage fever in 1996. As such, many workers in medicine have been awarded Nobel Prize for their outstanding contributions in microbiology. The methods of many infectious disease and vaccine production have been revolutionized. Example, recombinant DNA technology, PCR, nuclear antiprobes, radio immunoassay, ELISA, etc. Branches of microbiology. Microbiology is one of the largest and most complex of the biological sciences because it integrates subject matter from many derived disciplines. Microbiologists study every aspect of microbes, that genetics, their physiology, characteristics that may be harmful or beneficial, the ways they interact with the environment, the ways they interact with the host, and their uses in industry and agriculture. Each major discipline in microbiology contains numerous subdivisions or specialists that deal with the specific subject's area or field. Branches of microbiology. Branches of microbiology include: number 1, medical microbiology, industrial microbiology, food microbiology, microbial genetics, and molecular biology. Deals with the infection of genetic function of genetic material and biochemical reactions of cells involved in metabolism and growth. 5, microbial ecology deals with the interrelationships between microbes and the environment, the roles of microorganisms in the nutrients cycle of soil, water, and other natural ecosystems. Here we are concerned with the medical microbiology. It is studied under following headings. A, parasitology, deals with the study of parasite causing disease in human being. B, mycology, deals with the study of fungi causing diseases in human beings. C, immunology, and concerned with the mechanism involved in the development of resistance by body to infectious disease. D, bacteriology, deals with the study of bacteria. Genetics is the study of heredity and variations. Virology is the study of viruses. Scope of microbiology. Why we are studying medical microbiology? Number 1, diagnostic, example, isolation, and identification of causative organisms from the pathological lesions. We can also diagnose typhoid fever by doing Wedel's test. 2, prognosis of disease, example, in Wedel's test, rising titer significance, active diseases, and ineffective treatment. Falling titer means the effective treatment and curing of disease. 3, guidance in treatment, example, by culturing the organism in a pure form and then performing drug sensitivity tests, we can suggest the effective drug for the treatment of the particular infection. 4, source of infection, example, in sudden outbreak of infectious disease, we can find out the source of infection. 5, detection of new pathogens and then development of vaccines. Bacterial taxonomy. Bacterial taxonomy comprises of 3 separate but interrelated important areas. Number 1, classification. It is the arrangements of bacteria into taxonomic group or taxa in singular taxon on the basis of similarities or differences in their biochemical, physiological, genetic, and morphological properties. 2, nomenclature. It refers to the naming of taxa according to their characteristics by following the international rules. 3, identification. It is the particular side of taxonomy, the process of determining that a particular isolates belong to recognized taxon. The taxonomic system has three primary functions: classification, nomenclature, identification of species. The 8 major taxa or groups in the taxonomic system are in descending order: domain, kingdom, phylum or division, class, order, family, genus, and species. The binomial system of nomenclature describes each living organism by 2 names, genus, and species. Biological weapons. Now it is quite clear and indestructible that biological warfare is not new at all. Biological warfare was reported by early Romans who polluted water sources of their enemies by dumping animal carcasses. Then the British distributed blankets to Indians in 1763. These blankets had been used by smallpox patients. Hence, India user contracted smallpox. British had detonated an experimental anthrax bomb in Granard Island in Second World War. In 1984, 750 people fell ill to food poisoning in Oregona because of spread of salmonella that had been cultured in the laboratory. Biological warfare may be defined as intentional use of doses to harm or kill in adversary military forces, population, food, or livestock, and includes any living or non-living organism or it is bioactive substance toxin. Hence, germ warfare can be spearheaded by bacteria, viruses, fungi, toxin, etc. Organisms which can be used for biological war are Bacillus anthracis, causing pneumonia with a mortality rate 95% if untreated. Smallpox, contagious with the high mortality rate. Yersinia pestis, plague causing bacteria. Francella tularensis, tularemia. Ebola and Marburg viruses, hemorrhage fever. Clostridium botulinum, botulism, etc. Molecular epidemiology. Molecular epidemiology is defined as a science that focuses on the contribution of potential genetic and environmental risk factors identified at molecular level to the etiology, distribution, and prevention of diseases within families, countries, and continents. Genetic variability within and between infectious agents, disease pathogen forms the foundation of molecular microbiology. This table includes molecular epidemiology, ancient area, middle area, current area, or threat of rare diseases. In ancient area, smallpox, spread, plague, anthrax. Middle area, cholera, malaria, typhoid, tuberculosis. Middle area, emerging and re-emerging, cholera, dengue, leptospira. Current area, viral hepatitis, while HIV cause disease, acquired immunodeficiency syndrome. Threats of rare diseases, Ebola virus, yellow fever, or Rift Valley. Application of molecular epidemiology. Tracing, number 1, tracing the source and origin of infection. 2, tracking the routes of pathogen transmission. 3, identifying reservoirs sustaining transmission. 4. Identifying new emerging and re-emerging pathogens. 5. DNA fingerprinting in actual diagnosis of pathogens. 6. Characterizing drug resistance strain. 7. Monitoring the progress of diseases and central activities. 8. Identifying links between cases and infections. 9. Linking pathogen variants to endemicity and epidemicity. 10. Monitoring impact of immunization program, example polio eradications. Thanks for listening.